Hello everyone, myself Professor Shahana Sengupta. Presently, I am associated with Swami Vivekananda Institute of Modern Science, Kolkata as an assistant professor in the Department of Microbiology. I would like to welcome you all in my YouTube channel. In my YouTube channel, usually I share different types of academic videos related to the field of biological sciences. Today, I have picked up a topic for you all, that is biofertilizer. It is a very burning topic in today's scenario. Let's start our journey with a new topic. Happy listening. Let's discuss about biofertilizers. Biofertilizers are natural fertilizers which contain living microorganisms. When applied to seed, plant surface or soil, colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plant. Biofertilizer may also help in biological nitrogen fixation for the benefit of plant's body. Nitrogen fixation is defined as a process of converting the dinitro molecules into nitrogen compounds. For instance, some bacteria can convert insoluble forms of soil phosphorus to soluble forms. As a result, phosphorus will be available for the plants. They help in build up the soil microflora and thereby the soil health will be improved. Biofertilizer also include organic fertilizers or manures. Use of biofertilizer is recommended for improving the soil fertility in organic farming. Let's focus on the types of biofertilizers. Biofertilizers mainly are of three types, like bacterial biofertilizers, fungal biofertilizers, and algal biofertilizers. Besides these three major types, some aquatic fern also can be used as a potential source of the biofertilizer, and in some cases, with the help of the earthworm, biofertilizers also can be produced. Let's focus on bacterial biofertilizer. The live cells of bacteria are used as a biofertilizers. These microbes may contain unique gene called as NIF or NIF gene, which make them capable of fixing the nitrogen. The nitrogen fixing bacteria work under two conditions. Number one, symbiotic condition or free living bacteria, that is non-symbiotic. The symbiotic bacteria make an association with crop plants through forming nodules in their roots, whereas the free living bacteria do not form any association but live freely and fix atmospheric nitrogen for the plant's entity. Let's discuss about different important nitrogen fixators. I would like to start with Rhizobium first. Rhizobium is a gram-negative bacterium which inhibits the root nodules of most of the leguminous plants. Rhizobia can fix nitrogen after becoming established inside the root nodules. Rhizobia do not produce spores and are aerobic and motile too. Rhizobia maintains symbiotic relationship with legumes by responding chemotactically to flavonoid molecules released as signals by the legume's host plant. The name Rhizobia was established by Frank in 1889. Let's start with Azospirillum. It mainly present in cereal plants and inhibits both root as well as surrounding of roots, forming symbiotic relationship and increasing nitrogen fixation potential of the cereal plant. Azospirillum is recognized as a dominant soil microorganisms which can fix nitrogen in the range of 20 to 40 kg per hectare in the rhizosphere in case of non-leguminous plant such as cereals, millet, oilseeds and cotton. Considerable quantity of nitrogen fertilizer up to 25 to 30% can be saved by the use of Azospirillum inoculant. These species have been commercially exploited for the use as nitrogen supplying biofertilizers. Now I would like to discuss about free living or non-symbiotic bacteria. They do not form nodules but make association by living in the rhizosphere. Mostly they are present in the soil. Commonly used free-living bacteria are Azotobacter and Klebsiella. Let's talk about the Azotobacter. Azotobacter is a biofertilizer which provides the required amount of nitrogen to the plants from the soil. Azotobacter is a heterotrophic, free-living, nitrogen-fixing bacteria present in alkaline and neutral soils. Azotobacter is the most commonly occurring species in arable soils of India. Apart from its availability to fix atmospheric nitrogen in soil, it can also synthesize growth-promoting substances such as auxins, gibberellin, and also to some extent some vitamins. Many strains of Azotobacter are also exhibit fungicidal properties against certain species of fungus. Response of Azotobacter has been seen in rice, maize, cotton, sugarcane, pearl, millet, vegetables, and some plantation crops. It improves seed germination and plant growth. Azotobacter is the heaviest breathing organism and requires a large amount of organic carbon for its growth. Let's discuss about the process of mass production of microbial fertilizer in stepwise manner. This is very important in analyzing the future prospect of biofertilizer and its application. First of all, isolated bacterial culture were subcultured into nutrient broth. Secondly, the culture was grown under secking conditions at about a temperature ranges 28 to 32 degrees. Then the culture incubated until it reaches maximum cell population. Under optimum condition, this population level could be attained within 4 to 5 days for Rhizobium, 5 to 7 days for Azospirillum, and 6 to 7 days for Azotobacter. The culture obtained in the flask is called starter culture. For large-scale production, inoculum from the starter culture is transferred into large flasks or fermented and grown until required level of cell count is reached. After that, it is sterilized and cooled down. Media in a fermenter is inoculated with the log phase of culture grown in large flask. Usually 1 to 2% of inoculum is sufficient to obtain the proper growth of microorganisms. Cells are grown in fermenter by providing aeration and continuous stirring. Broth is checked for the population of inoculated organisms and cells are harvested with the population load of 109 cells per ml. Now I would like to discuss about the Algal Biofertilizer. Major two important algal biofertilizers are biofertilizers made up of blue-green algae and biofertilizers made up of Azolla. Now I would like to focus my discussion on cyanobacteria, or commonly called as blue-green algae. They are free-living nitrogen fixators. More than 100 species of BGA can fix nitrogen. Nitrogen fixation takes place in a specialized cell called heterocyst. BGA are very common in rice field. Unlike Azotobacter, BGA are not inhibited by the presence of chemical fertilizers. If no chemical fertilizers are added in a cropland, inoculation of algae can result in 10 to 14% increase in crop yields. They are very easy to produce. Usually, their mass production occurs in a cement tank filled with fresh water. No special processing is required. It is quite cheap. Cost of 10 kgs may be 30 to 40 only. It is beneficial for certain crops like vegetables, cotton, and sugarcane. Next important algal biofertilizer is Azolla. I would like to conclude my discussions after telling about Azolla. Azolla is a tiny freshwater fern commonly found in ponds, ditches, and rice fields. It has been used as a biofertilizer for a rice in all major rice-growing countries, including India, Thailand, Korea, Philippines, Brazil, and West Africa. The major nitrogen-fixing work is accomplished by the symbiotic relationship between the fern and BGA Anavena azolla. In addition to the nitrogen, The compost Azolla also provides potassium, phosphorus, zinc, iron to the crop. Azolla biomass gets doubled within 5 to 7 days by vegetative method. It can fix 40 to 80 kgs of nitrogen per hectare per year. It is a very good manure also for flooded rice. Increase of crop yield up to 15 to 20% has been observed while fertilizing the rice with Azolla. Hybrids are growing faster. It is also tolerant to heat and cold. Thanks a lot for your patience. Please do subscribe my channel. Stay tuned for the upcoming videos in different fields of biosciences.